Good morning, everyone. Today I will be discussing about manufacturing of antibiotics. Okay, so uh, I hope all of you had a very good puja holidays, and after the puja holidays, you are back to your normal schedule. Okay, so while I am teaching, if you have any problem in understanding anything, please feel free to interrupt me in between and you may ask the questions. Okay. So, because I will be taking only two of your classes, industrial biotechnology classes, so you may not get me later on. Okay, so I would prefer if you have any doubts in my lecture, you please ask me in between, interrupt me in between and ask me. Okay, so today, as I told you, I'll be discussing about um, manufacturing of antibiotics. Okay, so by the way, am I audible to you guys?
Yes, sir.
Yeah, okay,
Yes, sir.
yeah, good. So, uh, what do you mean by the term antibiotics? The term antibiotic, it has been derived from two words, anti and bios. Okay, the anti term it means against, and bios means life. So, if you look into the word, it means against life. Okay, as a whole, the this antibiotics terms it means against life. Okay. So, what are these basically? So, antibiotics, they are regarded as the chemical substances that are secreted by some microorganisms. This is an important thing. Antibiotics, they are chemical substances and they are secreted from some microorganisms. The chemical substances which these microorganisms secrete, they inhibit the growth and the development of other microbes. Okay. So, antibiotics are the chemical agents which are being synthesized by some of the microorganisms, and these chemical agents are used for inhibiting the growth and the development of the other microbes. Okay. So, classification of antibiotics. The antibiotics may be classified into two categories depending upon the activity spectrum of the antibiotics. Okay, the first one is the broad spectrum antibiotics. Okay, so the broad spectrum antibiotics, they act upon several unrelated microorganisms. Okay, as for example, tetracyclines and chloramphenicol. Okay, the narrow spectrum antibiotics, they act upon selected species of bacteria. Example, penicillin and streptomycin. Okay. So from here what we get? So the broad spectrum antibiotics, it will be acting against a large number of bacteria, and uh, in general these antibiotics, they are non-specific type antibiotics, and they are acting on a specific mechanism of the bacteria which is common on all the bacteria or the microbes. Okay, on the other hand, narrow spectrum antibiotics are the antibiotics which are acting only upon some selected species of bacteria. That means these antibiotics, they are targeting some activities. Someone's microphone is open. Please mute your microphone. Okay. So these narrow spectrum antibiotics, they target some of the specific pathways in the microbes which is specific to that special groups of the bacteria. Okay. Then secondly, the antibiotics can be classified depending upon their antibiotic activity. Okay, so what are they? They are antibacterial antibiotics, anti-tumor, antifungal, food preservatives, anti-protozoal, anti-tuberculosis, and anti-amoebic. Okay, so if you see, in all of these cases, we have some living organisms or living tissues which have been associated with. Here, antibacterial, they act against bacteria. Anti-tumor, they act against tumor cells. Okay, antifungal, they act against fungus, right? Food preservatives, this is a basically it is used for the what do you call for the preservation of the food, and it gives a it is basically a broad spectrum antibiotics. However, these antibiotics, they are edible, and if you consume these antibiotics, they will not create any side effect, or they will exert very minimal effect in the minimal or negligible or no effect when you are consuming it within the prescribed limit. Then anti-protozoal, these acts on the protozoa. Okay, anti-tuberculosis, these antibiotics, they act on the tuberculosis, mycobacterium tuberculosis. Then anti-amoebic, these acts on the amoebic against the amoeba and helps in amoebic infections. Okay. Now let us take the examples of these antibiotics. So antibacterial agents, we have penicillin G, and it again acts against penicillin species. Okay. Then cephalosporins, it acts against Acremonium species. Then streptomycin, it acts against streptomyces species. Tetracycline, it also acts against streptomyces species. Chloramphenicol, it acts against cephalosporium species. Bacitracin, it acts against bacillus. Then we have anti-tumor agents. So these, the first example is actinomycin D. It acts against streptomycin. In fact, all the anti-tumor agents, they act against this streptomyces species. And the examples of these anti-tumor agents include this actinomycin C, mitomycin C, actinomycin D, sorry, mitomycin C, bleomycin, adriamycin, daunomycin. Okay. Then we have the antifungal agents. In the antifungal agents, we have graciofulvin, which acts against, apart from the fungus, it also acts against the penicillin species. Okay. Then food preservative agents, we have natamycin and nisin. Both of these, they act against streptomyces species. Okay. Then we have antiprotozoal agents. Here we are having daunorubicin. It also acts against streptomyces. Then we have anti-tuberculosis agent, rifamycin. It acts against nocardia species. Then we have anti-amoebic agents, where we are having tetracycline and fumagillin, and these acts against streptomycin and aspergillus species respectively. Okay. Now coming to the manufacturing of the antibiotics. Okay. In general, the antibiotics, they may be synthesized or manufactured by three methods. First is fermentation. Second is fermentation followed by synthesis. This may also be regarded as semi-synthetic. Okay. And third one is chemical synthesis. Okay. So in the third group, we have chloramphenicol. Okay. In the second group, we have some classes of penicillin, like penicillin G. Okay. And in the first group we have most of the antibiotics. Okay, so today we will be discussing about mostly about the fermentation based manufacturing technique. Okay, so when we talk about this manufacturing of antibiotics, we have seen that we have discussed that the antibiotics are being synthesized by some microorganism. So the choice of the microorganism is the first and the foremost thing when you intend to develop some antibiotics. Okay, so what are the criterias which you should look into before you start manufacturing the antibiotics? The first thing is the microorganism what you should be choosing, it should have high yield of antibiotics. Okay, that means suppose you are having uh, two species, say species A and species B, both can synthesize some antibiotics, say AB. However, for a typical set, A produce only 1 gram, whereas B produce 10 grams of the antibiotics. So which one you will prefer? Definitely the second one, because the yield in the second one is higher. Okay, so the first thing what you have to check is what is the yield of the antibiotics when you use that specific microorganisms. Okay, why this is important? Because higher the yield, the more economically the process becomes viable. Okay, because if you are using say 100 rupees for both A and B, including the culture medium and all other setups, if you are getting 1 gram and if you are getting 10 grams, in the 10 grams you will be having a lot more of profit. So it will make your process more viable, economically viable, right? So you will be having more profit. You will be able to look into the process control in a much better way, right? And you can make better quality products, because when you talk about economics is also associated with your quality. So the more the money you put in or pump in into a process, you will get a relatively better quality product. It is not always true in all the cases. Okay, but in general, if you put in some extra money, then you will be able to get better quality products. Okay, and the third point, while you will be choosing the microorganism, is the most important point over here. Okay, this microorganism should not produce chemically related compounds. Okay, this microorganism should not produce chemically related compound. Say for example, you are synthesizing B, which is having a particular chemistry. Okay, so along with B, if the microorganism synthesizes B dash, which is a chemical analog of B, then what will happen? Then at the end you need a purified B. Okay, now during the purification process, if B dash is the analog of B, it will be very difficult for you to purify B from B dash, from a mixture of B and B dash, right? This is because since B dash is an analog of B, they will be having similar properties, if not the same. Okay, now if they are having similar physicochemical properties, then it becomes quite a difficult task to purify both the products. So you have to keep this in mind that the microorganism what you are choosing, it should not produce chemically related compounds of the products which you are targeting to synthesize. Okay. Once you have chosen the microorganism, the next is choice of the medium. Okay. So what should be the choice of the medium? So in most of the cases, corn steep liquor it is used. What is this corn steep liquor? Anyone who can tell me? What is a corn steep liquor? Anyone? None of you are there?
Sir, a corn strip liquor is a byproduct of corn processing industry, and it is full of nutrients like nitrogen and carbon.
Yeah, so what is it? How do you get that? You're exactly you're correct, but what is that?
Sir, when from starch is extracted from corn, then it is obtained from that process.
Exactly. Good. So during the extraction of the starch, this corn strip liquor is being obtained. So what happens is that when you uh, soak the corns into warm water, then what happens? Slowly they will swell, and during the swelling process, the starch molecules they get segregated and they remain over there. So after the harvesting of the starch molecules, we are left with a liquid thing, right? It is similar to the stuff. Suppose you are making some rice, right? So while making the rice, by boiling the rice, you are getting the starch. Okay, you get the rice product, and then you are left with some viscous material, which is basically uh, full of starch molecules. Okay, so if you remove the starch molecules, if you get the water, now that water it will also be having a good amount of the nutrients. And here we are talking about corn. In corn, it is having a lot of starch molecules, but apart from that, it also has a variety of nutrients. Okay. Now, due to the presence of this variety of nutrients, the corn strip liquor, it is one of the excellent what you call media for the synthesis of the antibiotics. Okay, it acts as a growth medium for the microbes, right? So microbes can grow, they can get very good nutrients from this corn strip liquor, and they can multiply in a better manner, and the harvesting of the microbes will be of very good quality. Okay, it's something like this. If you have a person and if you give the child, not person, if you give a child a lot of nutrients, he will grow healthy, right? And if you devoid a child of the nutrients, he will become sick. And if you look into the productivity of the child who has received good quality nutrients, his productivity will be much higher. The same goes with the microbes also. When you are giving them food which is full of nutrients, they will grow, and during the growth, they will generate the secondary metabolites like the antibodies in a which will be of better quality. Okay. Apart from the presence of the high quality nutrients, corn strip liquor they also have a good buffering capacity. Okay, they have a good buffering capacity. So in most of the cases, in general, for the manufacturing of the antibiotics, corn strip liquor it is used. Okay. Next, depending upon some the microbes, we may add additional sugars. Okay, normally lactose or glucose are added, and this is specifically done if you want to grow some fungi. Okay, then comes the nitrogen source. It is used occasionally. Okay, and what is the nitrogen source which is being used? Usually soybean and peanut, they are used as the nitrogen source for the medium for the growth of the microorganism. Now, in many cases, even though corn strip liquor, it is having a good buffering capacity. However, there are certain antibiotics which are pH sensitive. Okay, and if you change the pH drastically, the antibiotics may become inactivated. Okay, they may undergo degradation. So, under those conditions, additional buffering salts may be added into the medium. Okay. Then lastly, we may add some precursor so that we can improve the quantum of the yield. Okay. Say for example, if we are synthesizing benzyl penicillin, excuse me, then phenyl acetic acid, it is being added. Okay. So that the yield of the benzyl penicillin, it becomes much more easier. Similarly, phenoxymethyl penicillin, for the synthesis of the phenoxymethyl penicillin, we have to add phenoxyacetic acid. Okay, so the microorganisms which we use, they promote the conversion of the phenylacetic acid to benzyl penicillin and phenoxyacetic acid to phenoxymethyl penicillin. Okay, so a typical medium, it should contain all these components depending upon what you are trying to synthesize. Then comes the cultivation of the microorganisms. So in the cultivation of the microorganism, what you have? Inoculation of spores over a slope from the master culture. So whenever you go to the industry, you are given with you means the production unit, they are given with the master culture. Now from the master culture, the spores are taken and a slope is being made from there. I hope every one of you knows what is a slope, because all of you are from the biotech background, right? Okay. Now once you make the slope they are checked for the contamination and the variation. Okay. So contamination means whether other microbes are there or not. If they are there, if other microbes are there, or there is a variation in the genetic makeup of the microorganism, then the batch is immediately rejected, because if there is any variation, that then it may mean that it would what it would do is that it may not produce the antibiotics in sufficient quantity, so that would be a loss making process. Okay, also further, if you have some contamination, suppose the contaminant whatever is there, it produces another compound B, then what will happen? You will be having your target compound A which will be produced. Apart from that, you will also be having B. Now segregating this will be a very difficult process, and also over the course of time, if the contaminant microbe it grows pretty rapidly, then it will outgrow the original microorganism and you will be having a higher quantity of B and a very meager amount of A. So what is happening? You are making a system where you are getting a very low amount of your active components. Due to this, at the initial stage itself, we have to go for the checking of the contaminants and the variation in the microbes. Okay. Then after this check is done, the spores are then transferred to the culture flask. Okay. So to after the transferring to the culture flask, they are incubated with shaking. Why shaking is done? Can anyone tell me? Why shaking is done?
Sir, for proper distribution of the spores and the nutrients.
Yeah, proper distribution of the spores, good. Apart from that, the shaking helps in the dissolution of the oxygen from the environment. Okay, so due to these two reasons, the shaking are being done. Now, once you have sufficient amount of the inoculum, the inocular is transferred to a tank which is called seed vessel, and which is basically a small version of a fermenter. Okay. Now, in this fermenter, you have to blow air, okay, so as to maintain the dissolved oxygen level within the medium. Okay. So, to maintain the dissolved oxygen, we have to pass sterile compressed air, okay, which is blown through the medium through a specialized attachment which is called sparger, okay, which is called sparger. Now, I hope many of you have seen uh, when we pass compressed air through an aquarium. So what happens? At the top of the aquarium, you will be having frothing. Okay. Now, while we are blowing the air into the medium, the frothing is one of the major problem over there, right? Now, to prevent the frothing, we have to add anti-frothing agent within the medium, anti-frothing agent within the medium, which will prevent the phenomena of frothing. Okay, so what are they? Some of the common examples of anti-frothing agent is silicone fluids, vegetable oils, and octadecanol. Okay. Now, can anyone tell me why anti-frothing is important? What happens if you do not add the anti-frothing? There will be frothing. So what?
Sir, cell may be washed out with the froth.
Yeah, not washed out, because if it is a closed chamber, the cell will get entrapped into the froth and they will float on the top of the medium. So they will not have a sufficient chance to grow and to synthesize the antibiotics which we are producing, which we intend to produce, right? Now, next is sterilization. Um, the sterilization of the culture medium, vessels, pipelines, and air must be carried out at regular intervals before and after we have cultured a batch. Okay, so the fermenter, seed vessel, and the pipelines, they are sterilized by passing steam under pressure. Okay, by passing steam under pressure. You have to keep in mind over here, no additional chemicals are preferred, because if proper cleaning is not done, because cleaning of these mm, uh, or the removal of these chemicals from the whole setup, it becomes a very tedious and costly affair. So, steam under pressure, it is the most cheapest and the most effective way to undergo sterilization. Okay. Then culture medium, how you do it? Again, we can go for autoclaving. Okay. And lastly, we have talked about that we are putting in compressed air. Okay. Now, how the air is being sterilized? The air is sterilized through a sterilizing filter. Okay. So the air is passed through the sterilizing filter. All of you now must be knowing this laminar flow cabinets and all these things. So what happens? Within the laminar flow cabinet, we get sterilized air. How we get? Over there, we have a motor which pumps in air, which sucks in air, and it throws it through a HEPA filter, which is basically a sterilizing filter, and then the air comes out. Okay, so when we are using such kind of filters, it helps us in sterilizing the air. Okay, by getting rid of the microbes. When we are talking about when we are using steam under pressure, here we are getting the sterilization by killing the microbes, but for the air we are removing the microbes to sterilize. So there is a basic difference in the process in both these steps. Okay, then the fifth point is we have to accurately maintain the temperature of the medium. Why it is important? It is important because the temperature of the medium, it will increase because the microbes, they are taking up the nutrients. In the nutrients you have a sufficient amount of carbohydrates, and they are metabolizing the carbohydrates. Okay, and when we are metabolizing the carbohydrate, heat is generated. Now that heat, it results in the increase in the temperature of the medium. Okay, now high temperature, they may inhibit the growth of the microbes. Okay, not only this, the high temperature, they may destroy the antibiotics also. As for example, if you take the case of the penicillin, they are being developed at a temperature of 24 degrees centigrade, because the penicillin, it is very sensitive to temperature. A temperature higher than 24 degree can induce the degradation of the original product. Okay. Then the next point is isolation of the product. How do you isolate the product? First thing is mycelium is removed by a rotary filter. What is a rotary filter? I hope everyone knows. What is a rotary filter? Any idea? What is a rotary filter? No one? So a rotary filter is a class of filter. So suppose I am drawing a crude diagram. I am not good in that diagram. Okay. Okay. So you have a filter like this. You have a drum, and in the drums you are having holes like this. Okay. Now this drum is enclosed within another chamber where you have an outlet. So you put your media along with your mycelium and all these things into this one, and inside this bucket there is a filter just inside this which is stuck over the inner surface of this bucket, which is having perforations. Okay, now what happens if you start rotating it at a very high speed? Due to the centrifugal force, the liquid medium, it will start coming out into the larger drum, and from there it will be drawn out. Okay, so you will be left with the mycelium. Okay, so the mycelium is first removed by this rotary filtration. Your filtrate is being collected. Okay, then the antibiotic, they are obtained by pH adjustment. Each antibiotic, they have a particular pKa value. If you adjust the pH near their pKa value, the antibiotic, they precipitate out, and you can remove the antibiotics from there. So after you get the precipitated antibiotics, we can wash the substances unwanted substances from there by continuous washing using suitable solvents. Okay. Then next, we then extract the antibiotics with a solvent system. That means extract means if you are having say system A or compound A, which is your main this thing, and in within that we are having a contaminant B. Now system A, it is soluble in solvent say X, and system B is it is soluble in yeah, solvent Y. And also system A, it is not at all soluble in Y. Okay, it is not soluble in Y, and the system, the contaminant B, it is not soluble in uh, uh, say X, not soluble in X. Okay. So under these circumstances, we will be choosing such a solution, a solvent, which will be specifically be dissolving the main compound A. So we will be putting that solvent, and it will dissolve the solvent, and all other unwanted substances, they will remain over there, and we will just filter that out. In some cases, we may use chromatography and ion exchange techniques for the separation of the required antibiotics. This is being carried out in the case of streptomycin. Okay. Now next is purification. The purification is usually carried out by the formation of complex salt formation. Okay. Example, N-ethyl piperidine salt of penicillin is being made to purify the octane. Thereafter, it is being solubilized, followed by recrystallization to get the purified product. And lastly, the antibiotics, the salts of the antibiotics, they are dried under vacuum or freeze dried. And at a later stage, they are aseptically filled into the vials, aseptic vials, and they are closed and they are then marketed. Okay. So in this method, whatever I have discussed, this is a generalized method for the synthesis of any of the antibiotics. Okay, it is a generalized method. So usually, if you go to an industry, and if you find that if you look into the process, you will find grossly this whole procedure, these 8 steps, whatever I have talked about, it is being followed for the synthesis of the antibiotics. Okay. So in some industry you will find penicillin is being synthesized. In some industry you will find they are synthesizing glycofulvin. But in general, the process for the synthesis of any of these antibiotics, they remain essentially similar. Okay, with slight modifications, right? So as for example, I told you, in the case of this penicillin, we need a temperature of 24 degrees centigrade. But for another antibiotics, it may be 35 degrees centigrade, right? Similarly, when we are talking about the isolation, see, for the isolation of the streptomycin, we have to use chromatography or ion exchange techniques for the isolation of the product. Okay, but it might not be the case with the penicillin. So there will be some certain changes in the process depending upon the requirement, so as to get a better yield. However, in general, this whole procedure will be followed. Now, coming to the antibiotics and what are the microorganisms so which are used for the synthesis of the antibiotics. One of the common group of antibiotics is penicillins. Okay, so here we have 6 amino penicillinic acid, which is also regarded as 6 APA, 6 APA. Okay, 6 amino penicillinic acid. Now this is the chemical structure of the penicillin. Okay, and this group is the thiazolidine ring, and this group is the beta lactam ring. Okay, so penicillins and cephalosporin group of drugs, they are also regarded as beta lactam antibiotics. Beta lactam antibiotics. Because both cephalosporins and penicillins, they have this beta lactam group in common, and this beta lactam group is an important group or important ring to maintain the activity of the penicillins and the cephalosporins. So there are certain microbes which synthesizes or which produces beta lactamase enzyme, and this beta lactamase enzyme, it can break down this beta lactam group. Okay, now since this beta lactam group, it is important for the synthesis, for the activity of the penicillins and the cephalosporins. Those microbes which generates or which develops beta lactamase enzyme, they make these penicillins and cephalosporins inactive. In other words, these the microorganisms which synthesizes beta lactamase enzyme, they become resistant to penicillins and cephalosporins. Okay. Now the penicillin, it can be synthesized by two methods. The first one is the direct method. Second one is the indirect method. In the direct method, penicillin chrysogenum, it is used for the penicillin fermentation. Okay. And in the indirect method, phenylacetic acid, which is a precursor of benzyl penicillin, is added at different intervals to the fermentation broth. Later, E. coli is used to obtain the penicillin from benzyl penicillin, which basically E. coli releases acyclase enzyme, which helps in the conversion of benzene penicillin to the penicillin. In most of the cases nowadays, the indirect method is used because the yield by the indirect method is much higher than the direct method. Okay. Then the next antibiotic is streptomycin. The streptomycin, it is being developed or it is being produced from Streptomyces gracius. Okay. The drug streptomycin itself, it is highly unstable in nature, and due to the reason, ion exchange techniques, it is used for the purification of the antibiotics from the broth. Since streptomycin is highly unstable, you will find that the solutions of streptomycin are usually not available. Okay, usually even if you have to make the intravenous fluids or the intravenous preparation of the streptomycin, they are prepared as dry salts. Okay, and in the dry salt form, streptomycin, it is stable for at least 2 years when they are kept at a temperature of 30 degrees centigrade. Now separately, if you have to prepare the injections, then the injections, they have to be buffered between pH 3 and 7. Okay, and sterilization is carried out by filtration due to its unstable nature. Okay, you cannot go for heat sterilization or autoclaving. Okay, then next is tetracycline. So tetracycline, it is being produced by Streptomyces viridifacieans. Then chlorotetracycline and dimethyl chlorotetracycline, it is being synthesized by Streptomyces Oreoficians, okay, and lastly oxytetracycline. It is being synthesized or developed using the microbe Streptomyces remosus, okay. Then chloramphenicol. Previously, it was being synthesized through Streptomyces venezuelae. However, nowadays this antibiotic is being synthesized by synthetic method, which is much cheaper as compared to the this fermentation based method. Okay. Then we have erythromycin, which is synthesized or developed from Streptomyces erythraeus. Then cycloserine, it is being synthesized using Streptomyces orchidaceus and Streptomyces garifellus. Okay. Then neomycin sulfate, it is being synthesized from Streptomyces fradiae. Viomycin, it is being synthesized from Streptomyces griseus variety purpureum. Okay, purpureum. Then novomycin. Here, novomycin is being synthesized from Streptomycin niveus. Then graciofulvin, it is being synthesized using Penicillium graciofulvus. Okay, and lastly, nystatin, it is being synthesized from Streptomyces norsei. Okay, so this is it about the manufacturing of the antibiotics by the fermentation technique. So if you want anything, we can discuss. If you have any problem or doubts, are you guys there?
Yes, sir.
Yeah. So any one of you, do you have any problem? No? Okay then. I think we'll stop then.